Hello and welcome to our next chapter, which is chapter 11. This is animal physiology, so this is going to basically be like human phys. It is going to build off of knowledge that you should already have. So I know that you might not remember what was going on last year, but chapter 6, you can find that on our Google site. Chapter 6 is information that you should already know. This specific lecture is Defense Against Infectious Disease higher level. There is a base level amount of information that you should already know about this. So I'm not really diving into the base knowledge that you should have, but if you are going through this and you're like, what the heck are you talking about? Please go back to chapter 6 to fill in all the missing pieces. So let's get started with this. I do have a little bit of a review. These are just sort of like main terms that we're going to be talking about. So pathogen, any disease-causing microorganism, host specificity, right? Some pathogens are very specific to the species that they infect, and some of them are not. So zoonosis are types of diseases that can be passed from humans to humans from other animals. Think like rabies, right? You can get rabies from like raccoons or dogs, Lyme disease from ticks, West Nile from mosquitoes, tuberculosis from cows. But then some pathogens are mega specific, right? Polio, measles, syphilis is actually only a human disease. And then antigen and antibody. So an antigen is a foreign substance that causes a response, and an antibody is the response that your body produces to that antigen. Those two are going to come up very frequently. So if you are already like, whoa, those sound very similar. Yeah. Highlight, star, make these two known because you will be using them, and you do not want to interchange them on accident. So continuing on with antibodies, they're made up of 4 polypeptide chains, typically forming a Y-shaped molecule. The ends of the arms are what we really care about because they're typically very specific to the virus or bacteria, whatever it may be, that is infecting the body, because those can change depending on what the antibody is up against. Each antibody will recognize a unique antigen fragment, which is why the ends are so specific, because they are very particular to what diseases they can actually fight off. So let's dive into the immune response. So you have a first line of defense, which is like your skin, mucous membranes, right? Like your nose, even your mouth, right? These are just like basic, non-specific, kind of just like general defense, right? They're gonna prevent most things from getting in to your body. That's literally why you have skin. The second line of defense is phagocytic cells, antimicrobial proteins. This is like your inflammatory response. This is like if you get a cut, it's going to turn red, right? Your body is trying to fight off that, remove the infection, whatever it may be. This is like when you breathe something in, like dust or something, you will sneeze to get it out. It's just like a second line of defense. And then the third line of defense is specific. This is your actual immune system. This is dealing with your antibodies, your lymphocytes, your macrophages. These are like your big time cells that are involved with the immune system. So kind of talked about this, right? Second line of defense, increased blood supply. You're secreting histamines, which increase blood supply, capillary permeability, and attract phagocytes, all of which are great because it's a very non-specific response, and it's just like a sort of like a general, something is bad, I will attack, and hopefully that solves the problem type of response. Whereas the third line of your immune response is super specific. And these steps are important, and I know it's a lot of steps. We are going to talk about it quite a bit, but you do need to write this down, and frankly, you should probably memorize this. So when a pathogen infects your body, the first step is that it's engulfed or eaten by a macrophage. That takes on the antigen or epitope of that pathogen. So it produces an epitope, which is like a protein that sticks out on the surface of the cell, and that epitope is presented to T cells. And then the helper T cells are activated. And then the helper T cells stimulate the B cells. And then the B cell produces clones. And clones can either be plasma cells, which continue to produce antibodies in hopes of fighting off this infection, or the clones can become memory cells, and those memory cells stay in your body even after this pathogen has like run its course in your body. The memory cells stay so that if you get infected again, then the memory cells are like, whoa, I've seen this guy before, like, let's do this. And then the memory cells produce plasma cells that produce antibodies, and it's a faster response time. So this is literally the exact same thing as what I just said, but it's in picture form. So your star is your pathogen. It's ingested by the macrophage. It's digested and an epitope comes out, right? This like star point. This epitope is presented to helper T cells. And what do you know? There's a helper T cell that fits the shape, because the shape is specific, of this epitope that was just created. So then this activates the helper T cell, which activates the B cell. Once the B cell is activated, the B cell becomes clones and differentiates into the different types, right? Plasma cells produce the antibodies. You can see them being produced, and then they get shot out, and then they collect all of the pathogen parts and they destroy them. Or memory cells, which is the basis of immunity, really, but it really mostly helps if there is a second invasion of this pathogen. So, principles of antibody production: clonal selection and memory cells. Clonal selection is the B lymphocytes have a specific antibody on its surface that recognizes a specific antigen. That's like the specificity. That's why the immune response is not, the third line of defense is specific. It's not just a general response. The B cell with the appropriate antibody will become activated. So you have tons of B cells in your body, but only the specific one will become activated when infected with a specific pathogen. And then the B cell clones, plasma cells, produce antibodies, giving immunity. The memory cells are what gives you long-term immunity. They live on in the bodies even after the pathogen is gone. Maybe in you don't have like a million of them, you just have a few that live on, but it allows a rapid response. And that's the key here. This is why, like, if you get sick and then you get sick again a couple weeks later, it's not nearly as bad. This is also how vaccines work, which we'll talk about a little bit later. But in general, memory cells are awesome and help with fighting off diseases. Now, polyclonal selection, right? We've been talking about monoclonal selection, right? Where you have one antigen, one epitope coming out, right? So in this case, an antigen has different structural proteins, right? And there's multiple on here. So this antigen is taken in, it's ingested, but you can see that there's like multiple pieces here, right? The yellow circle and the pink triangle. Ingested by the macrophage, and then you present multiple epitopes on the macrophage, which means that you are activating multiple helper T cells, which means that you are activating multiple B cells, which means that you are throwing out multiple antibodies, and the multiple antibodies attack the pathogen at the same time. So it's the same general idea. It's just that when you get to the T cell step, you're just activating more than one, right? Poly means many. It all makes sense, hopefully. Now, there are many different ways that antibodies can destroy a pathogen. I'm going to talk about 5 ways. So there's opsonization, which just makes the pathogen more recognizable so it can be engulfed more readily. Neutralization, which prevent viruses from docking to other cells, right? Viruses need a host in order to reproduce. If they don't have a host, they are unable to do anything. The neutralization of toxins, right? Bind the toxins produced by they bind to the toxins produced by pathogens and then bind to the pathogen surface and cause it to burst, which is interesting. And then agglutination, which is sticking pathogens together so they can't enter host cells, and then phagocytes can more easily ingest them. And then lysis, which basically just act as like cell destroyers. To lyse something is to kind of like make it burst. So lysin's only job is to go into destructive pathogen cells and cause them to get destroyed. Now let's talk allergies. I don't know if you have allergies, but I am like basically allergic to anything that is outdoors. And allergies are reactions by the immune system, so they go through this like ridiculous immune response to items that are harmless. Pollen and bee stings, well, if you're allergic, can be detrimental to your health, right? They're very dangerous if you're allergic to them. But in general, they're not a pathogen, right? Your body is having a reaction, but normally they're harmless. So the substances that cause this allergic reaction cause overactivation of basophils and mast cells and an excessive secretion of histamine. So just so that we're clear, we kind of have a very similar situation here where the person who has allergies runs into an allergen. Like for me, I am allergic to literally grass and trees, so anything like that. I make an antibody for this, like pollen or whatever, and then my body reacts as if this is an infection, like a real honest to God infection from a pathogen. And so I am now suffering the symptoms, right? Wheezing, sneezing, my eyes are so itchy, like I can't do anything, because my body thinks that I'm under attack. So it's trying to help me. It's not, but it's trying to help me. Which is why when you take an allergy medicine, if you've ever like actually looked at the bottle, they're called antihistamines. And what it does is it prevents all of these histamines from forming, right? The secretion is slowed down, which means that your body isn't going through this reaction. That's how allergy medicine works. Okay, moving on a little bit, still talking about the term antigen, but now we're going to talk about blood transfusions. So blood groups are based on the presence of antigens on red blood cells. RBCs are red blood cells. The antigens stimulate antibody production in a person with a different blood group, okay? Which is why you cannot take blood from someone who has a different blood group than you, unless, it's very specific, right? Blood transfusions are heavily moderated because it can kill you, because your blood will literally attack the blood transfusion. So there are 2 main groups that we're going to talk about, the ABO blood group and the rhesus or RH blood group. The ABO blood group is probably what you're more familiar with, right? A, B, AB, O. This is just sort of like your general blood type. When people ask you what your blood type is, this is generally what they mean. You can donate safely to some, right? If you're O, you can donate to everybody, but if you are O, you can only receive O because those antigens that are on the other 3 blood groups are going to cause your blood to seize up, right? They're going to clump. It's going to get clumpy, which is obviously not ideal in your arteries and veins. So this chart is just basically a brief overview of whether you can donate safely or whether you can receive safely. You should be familiar with this. I'm not going to ask you to fill out this chart, but you should be familiar with a general idea of how blood donations work and blood transfusions work. So blood typing, this is, again, you're basically using the antibodies that are in your blood. You're like forcing them to react. And if you get coagulation or clumping, that's how you can determine the blood type. So the blood samples are mixed with anti-A and anti-B serum. That's the antibodies that you would normally find in A and B blood. And then if something coagulates or clumps, you can see that, and then you can determine what the blood type is. You do this to determine the ABO blood type and whether you are Rh positive or negative. So you should do this testing if you are getting blood from a donor or if you are doing a transfusion, of course. You absolutely, like, I cannot stress this enough. You don't want your blood to coagulate in your arteries and veins. That, I can't even imagine. That would be so painful. Now, Rh blood groups, that's the other type of blood group, right? ABO is like your general blood group that we talked about. Rh is you're either positive or you're negative. It's named for the presence of Rh antigens. You don't need to know anything else beyond you're either positive or you're negative. Most Americans are Rh positive. I myself am A positive. That's my blood type. But what's interesting is problems can actually occur, especially during pregnancy, if a mother is negative for the Rh factor, but the father is Rh positive. The child could inherit the Rh positive factor, and because the mother is negative and the fetus, the baby, is positive, they can actually send out those antibodies, and it can be super problematic because obviously you don't want the antibodies attacking the fetus or the mother, but if the baby can have a different blood type than the mother. So it's very interesting to see that. And it's important to do these blood tests prior to or at the beginning of pregnancies to determine sort of like next steps if this is going to be an issue, and how to take care of that. Okay, so blood clotting. We're getting to the end of this bit. So blood clotting, this general flow chart of how blood clotting works, you should know. You might be learning it for the first time right now, but you should know this and learn this. So when you damage a blood vessel, right? You've broken an artery, you're bleeding, you got a paper cut, whatever it may be. When the actual vessel is broken, it triggers the release of what's called clotting factors. There's a lot of things that make up clotting factors. I don't need you to know that. I just need you to know clotting factors. And when clotting factors are triggered, that makes prothrombin turn into thrombin. And when prothrombin turns into thrombin, thrombin activates fibrinogen, which is soluble, and that turns it into fibrin, which is insoluble. And once it's fibrin, that's how you get your blood clot, because the strands of fibrin actually connect to the red blood cells and they form these insoluble, obviously, clumps, and that's actually how a blood clot is formed. So the more you know. I lied. This is like the last bit, talking about immunity and vaccines. So active versus passive immunity. Active immunity is when you are exposed to the antigen through infection or vaccination, and you go through clonal selection. So your own immune system produces the antibody. That's active. Passive immunity is when antibodies are not made but are given. This can be transferred from another source naturally or artificially, temporarily protect against disease. So this could be done like during pregnancy or during breastfeeding, or an emergency treatment like if you were trying to get snake antivenom. Okay, I hope that you've heard that mothers when pregnant, they give extra protection to the baby. So a mother's antibodies can cross through the placenta and go to the fetus. Same with breastfeeding. When a child is like really young and still breastfeeding, the milk that is produced a few days after birth actually do contain antibodies and provide a natural sense of immunity to the fetus. Now, natural versus artificial. So natural immunity is the result of a natural infection, right? That makes sense. This is like the challenge and response method, which is like when you get sick, right? Your immune system is challenged by a pathogen and responds with a response like we've talked about at the beginning of this. That's natural immunity. Artificial immunity is broken down into two different things. There's active artificial immunity, which is a purposeful exposure to an antigen, like you get a vaccine. So you're purposely activating your own immune system. Whereas natural would be like if I was just out and about and I breathed something in unknowingly and got the flu, right? That's natural. But artificial is like making an appointment at the doctor's office and getting a vaccine. And then passive artificial immunity is when the antibodies are given to a patient, like after you're exposed to rabies, you get a shot full of the antibodies. So vaccination, this is a weakened or dead version of a pathogen injected into the body. You produce your B memory cells because you do go through that like challenge and response. It's just on purpose, right? You're not accidentally exposed to something. You're purposely exposed to this weakened or dead version. And then your memory cells go through the whole immune response, and then the antibodies stay as memory cells. So that means that, right, in here, like when you get the vaccine, you do have a response, and usually you might feel it. Maybe you feel a little like sick or just not as good as you normally do. And then if you actually get infected, the time of being able to hit that immune response is so fast, right? Your antibody production just shoots up because they already know what it is. They're expecting a second response, and so they respond quickly. This is active artificial immunity. Basically, this just prevents ill effects because the response is stronger, faster, and it's literally meant to protect you. The benefits, of course, there's a lot of benefits. Results in active immunity. You can avoid epidemics and pandemics, right? Smallpox was eradicated with vaccines. I mean, that particular, when we're talking smallpox, that particularly is successful because only humans can catch and transmit smallpox. So, like, when symptoms emerge, it's really quick that they emerge, and it's like readily visible. And the immunity of the vaccine is really long lasting. So while it's super awesome and like really cool that smallpox was eradicated, it is sort of like a special case just because like this, not side effects, but like the effects of smallpox are so particular. That is not the case for every single disease that we have vaccines for. Other benefits are, of course, preventing death, preventing disability like polio, and then a decrease in healthcare costs, right? If you have a vaccine, that means you're not getting sick, which means you're not going to the hospital, which means you're decreasing healthcare costs. Of course, this is particular to places where healthcare is privatized, like the US, but still, you do have a decrease in healthcare costs across the world, but this is particularly in like a particular benefit for people that pay for their own health insurance. And monoclonal antibodies, this deals with kind of how we make vaccines. So when you want to produce antibodies on a large scale, you produce monoclonal antibodies. And by doing this, you inject antigens into an animal, typically a mouse. So the antigen of interest injected into a mouse, which produces the B cells that we care about with the antibody on it, right? Then you fuse them with a tumor cell. And the reason that we do that is because cancer cells divide endlessly. They divide quickly and they never stop. So if we put that antibody with a tumor cell, that means you have an endless supply of these antibodies that are being created. And so once you get these cells that are producing like hundreds of antibodies, you can purify them, and then you're left with just the antibodies that you need that you can then inject into people for vaccines. Okay, so again, you do this for a lot of things beyond that, but you can neutralize toxins like anthrax, you can treat rabies. This is used for pregnancy tests. This is for cancer treatment, right? You target specific things. ELISA test, that's how you say that word, ELISA, to diagnose malaria or Lyme disease. All of these things are able to be done because we can single out the antibodies that we want. And last but not least, epidemiology, study of the distribution patterns and causes of diseases in a population. This data can be used to help plan vaccination programs. We use this data for like COVID-19 plans. And I'm sure you've seen, hopefully you've seen some data about this. Cases are monitored to find where further vaccination is required. Obviously, the ideal situation for an epidemiologist is to find areas of like high use of, or like a lot of high spread, a high infection rate, anything like that. Narrow your focus in on those areas and try and either get vaccines in there or get the spread down so that less people are being infected. Okay, so this was a long one. I know it was. I was envisioning that this was going to be long. If some of this is confusing, I urge you to go back to chapter 6, especially, I think it's 6.3, and get your basics of infectious diseases like under your cap so that you have a better understanding of this topic. We will dive into a totally different part of animal phys next time, and good job. See you in class.